Okay, I am recording. Yes. Okay. Now here, this is an investigation that involves respiration feast. And when you read the question, you will find that there is a layer of oil here that is 1 centimeter. Now, the moment there is a layer of oil, you know that this respiration is aerobic or anaerobic?
Anaerobic.
Anaerobic.
Anaerobic respiration. Oh, of course, you will read the question. Fine. I don't want to read it now because it will take a long time, but you will on the exam and you will decide for yourself what are the independent variables and what is the dependent variable. That's the first thing that you have to do. So the student investigated the effect of glucose concentration on the rate of anaerobic respiration. So from this first sentence, what is the independent variable? The
Glucose
effect of glucose.
concentration.
Yes, so you will take a pencil and you will underline it and you will say that this is the independent variable and the rate of anaerobic respiration, how much respiration happens over time as long as there is the word rate. So something or a certain measurement was calculated or divided over time. So this will be the dependent variable. This is the most important step before you continue. Okay, and then read the steps quickly. Label one test tube 0% and one test tube 0.5 and one test tube 1%. So this is the independent variable, the different concentrations of glucose. This is very good to remind me that when you do the investigation question, the other investigation question, when they ask you and you tell them that you have a particular independent variable, please give at least 2 to 3 examples. We have an investigation question in this exam. For example, if the independent variable in the other investigation question is temperature, don't just say, I will change the temperature. No, give at least 2 or 3 examples. Example, the temperature will be 50, will be 100, will be warm, will be hot. You have to give examples. So they're giving you here the 3 examples. Step 2, put 5 centimeter cubes of water into the test tube labeled 0, and then put 2.5 of 1% glucose solution and 2.5 centimeter cube of water into the test tube labeled 0.5. So they're showing you the steps. They're diluting their solution as we are going along. And then step 4, put 5 centimeter cubes of 1% glucose solution into the test tube labeled 1%, and then stir the contents of the beaker containing the yeast suspension with a glass rod. Step 6, add 5 centimeter cubes of yeast suspension to each of the test tubes labeled 0, 0.5, and 1. So this is definitely a control because we put the same volume of yeast in every test tube. Step 7, put all 3 test tubes into a water bath at 40. So this is another control. This is how I want you to read the investigation. As you are going along with a pencil, mark the variables, the control, so that when you come to answer questions, you will find what you want quickly and you will not waste time. Step 8, start the stop clock and wait for 3 minutes. So this is the time here in minutes. After 3 minutes, remove the test tubes from the water bath and place them in a test tube rack. Use a pipette to add 1 drop of methylene blue dye to each of the test tubes and carefully mix the contents of each test tube with a glass rod. And then the last step, use a second pipette to slowly add a layer of oil to each of the test tubes, of course, so that we block out the air. And the layer of the oil will float on top of the yeast, just like you see here. And we will wait until the methylene blue changes color from blue to colorless. We have this investigation. We took it somewhere. And then 12, put the test tubes back into the water bath. And 13, measure the time taken for the blue color to disappear. Okay, and they're giving you here the stopwatches and the time. Prepare a table to record the results of the investigation. Of course, you are all amazing in making tables, but still, I see some mistakes. And the common mistake that I'm seeing is that you put the units in the body of the table. No, no, no. We do not put the units in the body of the table. Now, quickly, tell me the concepts of a table. What will be put in the left side of the table? If this is a table with at least 2 columns, and this is the left side, which variable here will be on the left side? We're talking here in general about any table. Which
Independent.
variable? Very good. The independent variable. And that case will be the concentration of glucose and the units. The units here, you will not make it up. You will go get it from the question. So the unit here is in percentage. So in percentage, that's it. I know I said it 100 times, but I still see the same mistake. Don't write 1%, 0.5%. No, the percentage or the unit is already at the header. You do not put the percentage in the body of the table under any circumstances. And then what is the dependent variable? time. And here they were nice and they told you to convert the time on the stop clock to seconds. But this is not common. They do not tell you to do that most of the time. The time, the standard unit is seconds. So you will put it here in seconds and you will convert the time to seconds and you will put your answer here without units. Now, I know I have to ask this question because of some of you, not all of you. 1 minute is how many seconds?
60.
60. Okay, I know this is very childish, but I still see the same mistakes. Now, state, of course, you will do it here. I will not waste time doing it. State a conclusion for the results. Now, logically, the more the concentration of glucose, what will happen to the rate of the reaction?
Faster.
It will be faster, so it will take less time. So you will write it clearly here that the higher the concentration, the shorter the time for the methylene blue to change color, or in other words, the faster the reaction. State the independent variable. Now, there's
Concentration of
nothing easier
glucose.
than that. Exactly, glucose concentration. State one variable that was kept constant. Clearly, it was kept constant in this
Temperature of
investigation.
water bath.
Yes, temperature, it was kept at 40 degrees. Explain why it was important to stir the yeast suspension. Why did we mix? One easy answer.
To prevent
For
contamination.
no, no, this is not an answer. To prevent contamination, you will say this when they tell you why did they change the glass rod? Why did
طب
they
لسه
sterilize the
ممكن
here?
نشوف السؤال.
Okay, you will see the mark scheme.
No
Here, they
هو
are
بيسأل
stirring. Why
هو
is it
بيسأل
to stir the yeast in step 5? Why did we
to
stir? So that
activate it.
we, any reason, any reason. I can't hear you. I don't know why you're cutting off. But when you mix something, you mix it for even distribution of heat so that you do even distribution of the yeast. So the yeast does not settle down, so that you do even distribution of the nutrients, so that you start the reaction, you activate it. But the contamination, usually contamination means we are cleaning something or we are not using the same apparatus. We are changing the apparatus. So I will not use the same glass rod to prevent contamination. I will clean the apparatus to prevent contamination. Okay. But when I mix it so that I distribute. Always when you mix something for distribution to activate the reaction so that the particles, if they are heavy, they do not settle down. This is always the answer. Okay. Maybe if you give them this answer to prevent contamination, they will be nice, but I would not recommend it. Use contamination when it is related to anything got to do with cleaning or changing the apparatus. I will not use the same syringe. I will not use the same glass rod to prevent contamination. Okay. If I will mix to distribute the heat, to distribute the food so that the molecules do not settle down to start the reaction or to activate it. This will be my answer. Clear? Who was asking? I hope this is clear now. I'll highlight this. Okay, one way to improve the investigation would be to use an increased number of different concentrations of glucose. Suggest two other ways to improve this investigation. Now, please do not give the same answer. So don't say, I'm going to use different concentrations of glucose or smaller concentrations of glucose, because I see that they already gave you this as an option. So you need to think of other ways to improve. Don't write this answer. Give me other ways to improve any investigation. Give me answers quickly.
repeat and take average.
Of course, I'm going to repeat and I will take average. What else? I want another one.
ممكن نقول thermostatically controlled water bath.
But they did this in the investigation. They said that they put it in a water bath at 40 degrees. They did not say thermostatically controlled, but they did say that they tried to maintain the temperature. You can add that, but I want more ideas. How can you improve any investigation? Give me, you tell me.
repeat with a wider range.
Again, this is B. They repeated with different concentrations of glucose. They did not say smaller or larger, but they changed the concentration of glucose. So don't use this as an answer. Think of other answers. You have a list of modifications, a list in the practical part at the end of year 9 notes. Did they say in this investigation that there was a control? Did they? No, they didn't. So we can use this as a modification that we can design our own control. In this investigation, they used yeast. Yes? What can be your control? Think of controls. There can be one very important control. So we can design another investigation with a control to compare the results and give examples. What
ميس
can be
مش
your
هو
control?
الكنترول مش هو الكنترول الزيرو percent جلوكوز
Yes, but there can be another control.
without yeast.
Yes, with no yeast. But because you have to think of answers, my dear, you have to write here in number 2 something. You will not leave it empty. Okay? Because in this type of control, you will prove that the yeast is needed for the reaction. Okay? You have to give an answer. So we said we will repeat and we will calculate average. We will design another investigation and we will use a control, a different one from what they used. What else? What if there is a third here number asking you for a third way to investigate? What will you say? There are things that are standard. We can remove the anomalous results. Yes? Anomalous results and do not include it in the investigation. Here they used methylene blue. What if the color um, was not very obvious? Let's
White tile.
change the
Yeah.
method. Excuse me?
Use a white tile to identify the color.
Yes, we can see the color with a white background using a colorimeter. Then you will get better results. Or you can actually change the investigation altogether. And instead of measuring the time needed for the methylene blue to change color, you can, for example, measuring the amount of gas that is produced over a certain period of time using a gas syringe. You have to think of ideas. That is in the case they give you more than 2 suggestions. More than 2. Okay, but these 2 are very enough. I'm going to repeat. I'm going to calculate average. I can design another investigation with a control. I can calculate the volume of gas produced over time. Anything, but you have to think of answers. Okay, I will remove my anomalous results from the average. Okay. Let's look at the mark scheme. What other ideas do they give us? I can use a colorimeter. I can, what was the question? 1B. Okay. You will repeat at every concentration. We said it. Okay, so the first thing that crossed our minds actually is that we will repeat at different concentrations. So we said that. We will change the method to identify the endpoint. Example, we will compare against a white background, or we will use a colorimeter, or we will use a color chart or a color standard. We said it also. Do different concentrations at different times individually. In other words, this is not the same answer as we will do different concentrations of glucose. No, we will do the experiment, every concentration separately. In the experiment here, they did the concentrations or the different glucose concentrations at the same time. In other words, they had 3 test tubes at the same time, and they added the methylene blue to every test tube. And the 3 test tubes, they were observed at the same time to see the time needed for the color to change from blue to colorless. Maybe you will not concentrate, this is you and these are your eyes. Okay, you will not concentrate with the 3 test tubes at the same time. So it is better to do every investigation separately. This is another idea. Okay, so answer anything or say anything except changing the concentration of glucose, because this is already given. I do get those answers. Just like one of you, she said, I'm going to use bigger concentrations. No, this is wrong because this is already given. And usually when we repeat to improve, we use smaller concentrations, not bigger concentrations. Okay, so whoever gave me that question, please understand that you will not give an answer that is already provided. Think of other ways to modify. Are you with me? So we already said everything in the mark scheme, and we even said more. We said we can design a control. We will improve how we obtain our results, a colorimeter, a white background. We can measure the volume of the gas produced over time. We can do the investigation, each one on its own, remove anomalous results while we are calculating the average. Just you have a list of modifications because this question is repeated in every paper 6. Okay, let's not waste time. Describe how would you make 5 centimeter cubed of 0.25% glucose solution using a 5% glucose solution and distilled water. Now, this is a question, this is critical thinking. First, you will read the question and you will tell me I don't understand because I received a lot of students sending me on WhatsApp over this question. Maybe around nine students asking me about this question. You have to expect that you will get something different on every exam. You have a glucose solution that is 0.5%. We want to reduce this concentration to 0.25. So what are you doing to this glucose solution? You will increase in its concentration or you will dilute it.
Diluted.
You will dilute it. When you want to dilute anything, what do you add to it? I want
Water.
everyone to participate, not only Selma. Muhannad, Lean, Jolly. Yes, Selma is correct. If I have a concentrated solution and I want to make it less concentrated, I will add water. That's it. Now, the concentration of the glucose is zero point 0.5, and now I want it to decrease to 0.25. So here the concentration will be halved. Like, I'll give you easier numbers. If the glucose was 100% and I want to dilute it to 50%, what do? You will add water, just like Selma said. How much water? Randomly? Mm, lean. Muhannad,
Miss, you add 50% of water.
excuse me, my dear.
You will add 50% of water so that it becomes
Yes, you are
50%
correct.
and
Half
50%.
water, half glucose. I want to dilute it by 50%. I will add half water, half glucose. So all those 9 students that sent me over this question, they just did not notice the concentration decrease. They did not notice that it was 50% glucose and it has been decreased to 0.25, half half. So the final volume should be 5 centimeter cubed. Half this volume will be glucose and half of it will be water. So let me change color here. So what is half of 5? 2.5 centimeter cubed will be water, and 2.5 centimeter cubed will be the glucose solution. So I will have here a test tube that is half glucose and half water. So I will dilute my glucose, half. That's it. Okay, it's very easy, but don't panic when you read a question that is new to you. Just read it again. Try to understand the question again. Paper 6, you are not allowed to score below 35. The maximum number of marks that you should lose in this paper, of course, I prefer that you don't lose anything, but only 5 marks. Less than 5 marks, there will be a complete drop in the letter grade. So I want you to score 35 and above. Please. It's very easy. You can do it. Describe the method you would use to test a solution for the presence of glucose. Now, this is so easy. Yes, quickly tell me. What is
Benedict
the test
solution.
for glucose? Yes, of course, you will write Benedict. Of course, please use correct spelling because I see horrible spelling. This itself is 1 mark. And then you will describe it quickly. You will heat. Okay, in Benedict, you can even make a nice drawing. Here you have the Benedict and you have the glucose, and in a water bath that reaches at least 70 to 80 degrees. And you have to mention the color change, the starting and the final. The Benedict is blue and it will change color gradually until it finally reaches orange red. I expect you to know this very well. Okay, now,
Miss
describe suitable. Yes, my dear.
هو أنا لما أكتب الcolor change أكتب اللي هو blue yellow green ولا أكتب من blue gradually to brick red ولا orange red؟
They're both correct. In
Okay.
all cases, you just need to mention the final, the starting, and the final.
Okay.
Okay, describe suitable apparatus that could be used to collect and measure the volume of carbon dioxide produced. Okay, now this is also so easy. Here they said that they will change and they will measure gas. When you measure any gas, what is the apparatus?
Gas syringe.
Very good. A graduated graduated gas syringe. And again, I'm going to exploit the situation. Anytime you measure the volume of gas, if this is your dependent variable, I'll measure the volume of gas over a set time. I will not just leave the gas to be produced forever. So I will use a graduated syringe, or there is another way, an inverted graduated cylinder over water. Okay. And you can draw it also if they ask you in another exam, they were asking you to draw it, okay, and show that it is graduated. Very easy question. Now, what is an anomalous result?
A
A
result that
result
is
that's
different.
not with the pattern.
Yes, a result that is different from the rest. Now here, which result is anomalous? 1.8 or 3.2 or
2,
2 or 1.9?
3, 2, 2.
Yes, it's very obvious that it is the 3.2. Describe how the student calculated the average. When you calculate the average, do you include or you
Sum,
remove the anomalous results?
remove.
Remove the anomalous
Yes,
result.
of course, Selma, you wanted to tell me I'm going to add the sum and I will divide over the total. Yes, this is not the answer, my dear. They're not asking me how do I calculate average in general, but they are asking me how this particular student calculated the average. And then they are helping you with the answer because before they ask you this question, they asked you, what is the anomalous result? So they wanted the answer of, I'm going to calculate the average by dividing the total over the sum, yes. But while I will do that, I'm going to remove the what, my dear?
Anomalous. أنا كنت هقول
Very good.
excluding the anomalous,
The two.
calculating the average والrule.
Yes. Okay. I have to exclude the anomalous result. Okay. Now the table. No, still. Using the data in table 1.2, compare the mean volumes of carbon dioxide produced at 25 degrees and 35 degrees. Now, this question, it's got nothing to do with how much you studied at all. You will simply compare. You will only compare and you will not explain. So you will just take numbers. You will go to 25 degrees and you will say the mean volume of carbon dioxide produced is X and Y. And then at 35 degrees, using the data in table 1.2, compare the mean volumes of carbon dioxide. And where is 35? 35 is definitely somewhere else.
مس هو التيبل حضرتك
Huh?
كنتي على التايم.
أنا
هو
كنت على التايم.
التمبريتشر كانت في الليفت
This is
الرايت
because
سايد.
this is because, إيه temperature عشان I'm running.
أهو produced in
Here, صح
25 و
صح صح.
24.
Shatura. Okay, I'm going to compare this and compare this, and I'm going to see how the carbon dioxide is produced. Of course, it's very obvious that 35 degrees, the mean volume of carbon dioxide is increasing rapidly, and put numbers. Okay, here, you will say that it was constant, it was 0 and 0, and it did not increase even after 10 minutes, and then it started to increase slowly, and then it dropped again to 0.5, and then it increased again. So just compare the numbers and break them down. At 35 degrees, it was 0.1, and then it dropped to 0.8, and then it increased steadily. And in the last 5 minutes, it remained constant. Okay. So, no, I'm not going to rush here. When you come to compare, don't rush while you are comparing like what I'm doing. You are going to break it down. So you will say with 25 degrees, and please answer it nicely and in an organized manner. Why don't you do this? 25 degrees Celsius, you will do your comparison. And then at 35 degrees Celsius, you will do your comparison. Why not? It's nice and easy. At 25, it's very obvious that in the first 5 minutes, it was constant. There was no increase in carbon dioxide produced. And then there was a small increase. And then it dropped again. So here is another drop. And then it increased again. And write numbers. At 35 degrees, we started immediately. There was an increase in the carbon dioxide produced in the first 5 minutes. And then there was a rapid increase. And then in the last 5 minutes, it stayed constant. So there was constant here and then constant here. Okay, so this is how you break down your results. Now the graph. Plot a line graph on the grid of mean volume of carbon dioxide produced against time. Use all of the data in table 1.2. You will need to plot the data for each temperature as separate lines on your graph and include a key. Now, here you are going to plot two things on the same grid. Where is your independent variable? You will plot this table. So what will you plot on the x-axis?
Time.
Of course, it will be time. You will write it just as it is. I know that you are very used to that, but let's revise together. Time in minutes. Okay. And then you have what will be on the y-axis?
mean of volume بتاع CO2.
Exactly. The mean volume of the carbon dioxide, CO2 in centimeter cubed. I think this is the unit. So this is the first thing you will do. You will put a 0 here. Okay. And what will you do next? You will divide your graph into boxes of 10 or boxes of 5. It's nice and clear here. And you will use a key because you will plot two grids on the same graph. And then they said line graph. So please don't improvise and do a bar graph. I don't know where did you get this from. Now, as long as there will be a key, you will put a box here and you will make two graphs, one with X's or a solid line, one with a circle inside a circle or a dashed line, whatever. There just has to be any difference. And you will write clearly that one of them will represent 25 degrees and the other one will represent 35 degrees. Or you can write it on the line itself, whatever. Everything is accepted. Good. Now, we will not plot the graph one by one now, but we will distribute the values. You will go to the time, which we said will be plotted on the x-axis, and we will look at the maximum value. The maximum value is 30. So we'll go to the graph. We have here 1, 2, 3, 4 boxes. How can we distribute the values so that my 30 is at the end of the x-axis? So that my 30 is here. What do I do? That's very easy. Half of 30 here.
هيبقى 15.
Yeah, that's the easiest. This means that every 10 boxes is 7.5. Am I right? Okay, so 7.5, 15, 15 plus 7.5, you will have a value here. Let me get my calculator so that I do it with you. 15 plus 7.5, this will be 22.5. Some students also sent me pictures and they did this. What did they do? They did this. 5, 10, 15. No, they did 10, 20, 30. And by the way, this is not wrong. This was also accepted because the final value 30 took more than half the grid. It is, of course, preferred. And when you do it like this, when you distribute the values like this, it is easy to plot. So when the 30 was here, it is also correct, also accepted. And the values were easy to plot because every small box is 1. So this is good. This is okay. Or if you want to use the whole axis, you can do it like we did with the 15 in the middle and the 30 at the end. Both of them are correct. I'm making it clear. Both of them are correct. So this is how you will distribute the x-axis. Now, you will distribute the y-axis. You will look for the final value. The final value is 3.2. Let's say 3.5. I like to make it in 5. How many boxes do you have? 1, 2, 3, 4, 5, 6, 7, and 8. How will you distribute that? 1, 2. This is 1. This is 2. This is 3. This is 4. I see that this is the best way to distribute your values, and 3.5 will be it. This is so easy. Okay? Here, every 2 boxes will be 0.1. Did you do this graph? Did you plot this graph? Okay, I'm not going to plot it now online. It will take an hour on itself because it's very difficult to plot online. Do it, please, and send me the final graph. Okay? And you will plot both graphs. Of course, it's very obvious that with time, the mean volume of carbon dioxide will increase. So in both graphs, like in the first graph, the 25 degrees, it will remain constant and it will start increasing at 15. So in the first graph, it will remain constant like this, and it will start increasing at 15. So I expect it to look something like this. In the second graph, it will increase immediately from the first 5 minutes, and it will keep on increasing. So in the second graph, this is the first graph at 25 degrees. And from the second graph, it will start increasing from 5 minutes. It will look something like this. So please do it and send me your final graphs. Estimate the time taken to produce 3 centimeter cubed of carbon dioxide at 35 degrees. Show on the graph how you obtained your estimate. So if you do not show on the graph, you do not get a grade, even if you give me a correct estimate. So how do you show on the graph? After you finish your graph nicely, you will go to the graph and you will go to 30. Estimate the time taken to produce 3 centimeter cubed of carbon dioxide at 35 degrees. You will go to the graph and you will go to the time. You will go to what? 35. Estimate the time taken to produce 3 centimeter cubed of carbon dioxide at 35 degrees. How will you do that? Give me an answer. Hello? We want to estimate the time taken to produce 3 centimeter cubed of carbon dioxide at 35 degrees. You will go to 3 centimeter cubed here. You have two graphs. And you will move vertically until the graph of 35 degrees stops you. And then you will move vertically downwards. And you will be able to estimate the time. But you have to show it on the graph. Are you with me? Did I lose connection? Hello?
We are with you, Miss.
Did you understand how we got it? Estimate the time taken. So they want to know the time that is needed to produce 3 centimeter cubed of carbon dioxide at 35 degrees. So you will go to 3 centimeter cubed of carbon dioxide, which is here, okay? And you will move vertically until the graph stops you. Which graph? You have two graphs, one for 25, one for 35. So which graph should stop you? The one for 35 or 25? Give me an answer. I'm speaking to myself. Girls. Hello?
Yes,
Omar, why
Miss.
aren't you speaking? Do you know how we did it? Do you know how we got the answer
Yes.
here? Okay, it's
Yes.
very easy. Give me a reply. We're nearly finished. Carbon dioxide gas was bubbled through hydrogen carbonate solution and the indicator was red before the gas was bubbled. State the color change that would occur. When the hydrogen carbonate indicator, it was red before the gas was bubbled, and then the carbon dioxide gas was bubbled. What is the color change that would occur in the hydrogen carbonate indicator when it is acidic? What is the color?
Purple.
Purple? No, it's not purple. When it is acidic, what color will it be?
Blue.
Blue. Well, this is which number? This is D. This is 1D or 2D? Going to be yellow. I'm very surprised from this question because hydrogen carbonate is not core. Hydrogen carbonate is only supplement. So I'm very surprised that this question came in paper 6 because paper 6 is both core and supplement. Hydrogen carbonate, when it is basic, it is purple. When it is alkaline, when it is acidic, it is yellow. And in normal concentrations of carbon dioxide, it is orange. Please remember those colors. You will find those colors or this investigation in both year 9 and 10. Okay, so hydrogen carbonate, when it is acidic, it is yellow. When it is alkaline, it is purple. And when there is average or normal concentrations of carbon dioxide, it's orange. Now, this was a surprise to me because they usually never ask about hydrogen carbonate like this. They usually ask about lime water. So I'll highlight it also. Is this exam finished? No, we're only in question 1. Question 2. Figure 1.2 shows epidermal cells from a red onion and the magnification. Draw a large diagram of the cell labeled X. Now you have 4 marks for this drawing, and please do not ignore the drawings. I cannot ask you to draw it now, but we will draw X. Now, please pay attention to details. You always lose marks. What do I mean by pay attention to details? I mean, if the shape of the cell looks like this, then I will have to draw exactly the same shape, because I get students doing this. You will not get the grade because the outline of the cell is not the same. Even if you are used that plant cells are rectangular, this does not look like this. So you have to draw exactly what you see. So we do it quickly. I'm going to draw what I see. Number 1, take it as a rule. Anytime you draw a plant cell, please draw the cell wall as a double line. Double line, okay? So this is a plant cell. I'm going to draw an outline. I'm going to make it smaller so that I have more space. So I'm going to draw it like this, like this, like this, like this, and like this. Okay? As much as I can, make it as close as possible. And then, because this is a plant cell, the cell wall is a double line. And try to not get my pen out of the paper so that I have no broken lines. Of course, it's not empty. I can see a shrunken plasmolyzed cell. So I'm going to draw here this shaded area with the nucleus, and I can see protrusions here. So I'm going to give this exactly the same details. So I have a bulge here, if you can see. So exactly what you can see. We have a bulge here, then goes like this. I have a protrusion here, a small protrusion here. Of course, my drawing is not very good on the screen, but I'm just trying to tell you to keep the details. Okay? And a nucleus. No shading, no coloring, nothing. I tried as much as I can to keep it. And of course, this is small. I'm going to use more than half the space available. So I'm going to draw it here, and I'm going to draw the cell as big as possible. Big. Okay, use the space that is provided. Don't they give you all the space and then do something that is this small? So 1 mark for size, 1 mark for outline, another mark for details. Don't do any shading, no broken lines. Line PQ represents the length. Measure the length, and the length here in millimeters. So you will measure this length. You will measure it in what, and how do you convert it to millimeters? Quickly. How do I convert this length to millimeters? If I, for example, measure it, it is 3.5 centimeters. How do I convert it to millimeters? Give me an answer. Selma? Lean? Hello? Muhannad? Girls? Omar, are you there? Okay, I'll answer it for you. I don't know why your microphones are switched off, but you will convert it to 10, and this is going to change it from centimeters to millimeters, and then use the formula to get the magnification. Now, the investigation question. Water moves into and out of the cells by osmosis. Plan an investigation to determine the effect of the concentration of the sodium chloride on osmosis in plant tissue. This is slightly different from what you're used to, because the dependent variable here is something that is very used to you. But you will decide for yourself and write the independent variable will be the different concentrations of sodium chloride. And when you mention different concentrations of sodium chloride, you will give at least 2 or 3 examples. For example, 20%, 50%, and 90 or 100%. The dependent variable will be how this will affect the osmosis in plant tissue. Either you will measure the size of the plant before and after, or you will take a cell and look at it under the microscope. You have to think of a way to measure how will the change in the sodium chloride affect the plant tissue. We will use the transitional potato. This is plant tissue. We will use the traditional potato and measure its initial mass and then final mass and then the percentage change in mass, because this is something we are already used to. And then you will measure your steps. I'm going to get potato of the same size and the same type, different pieces of potato. And I'm going to start by measuring the starting mass of every potato. I will put it in this concentrated solution of different concentrations for 30 minutes. So I'm going to make the timing also constant. And then I will get the potato out of the solution. I'm going to dry the potato and measure the final mass for every potato. And for every potato, I will measure the percentage change in mass. And I'm going to get my results, which is going to be the different concentrations and the percentage change in mass when the potato was put in different concentrations for the same amounts of time. So your constant values will be the same type of potato, the same time, the same apparatus, the same volume of water that is put in every potato, the same temperature. All these are constant values. That's it. And you will write the last two sentences that is common, which is repeat at every concentration and calculate average. And for safety precautions, you can say that you will wear gloves or you will stay away from the concentrated solutions. This is the common investigation question. So this is a common exam, and the only thing that was a little bit new to you was the dilution. How can you dilute the glucose? You need to do a little bit of thinking. And when you dilute something, you add water, 50 solution. And the hydrogen carbonate indicator, they asked about it. Please memorize the colors, that it is yellow in acidic solutions, purple in alkaline solutions, and it is orange in normal carbon dioxide concentrations. Okay, best of luck and do the same. If you still have questions, send me the questions on WhatsApp and I'll answer you immediately.